So the odds ratio actually for our BRCA for breast is about 6. For ovarian, it's about 20. And I think the other thing that's important about the FH story is that the paper that you published that showed that your risk with a monogenic LDLR mutation really varies depending on your LDL cholesterol level. And that, while 3 is at the low end, if you have a relatively normal LDL cholesterol, if you have an elevated LDL cholesterol, it goes all the way up to 20. And so I think, we're at a point where we are getting to a point of equivalency for certain select cases, but to apply that as a general rule, I think for a number of the reasons that have been brought up is a little bit problematic at this point. Now, I did have a question for Naomi. And I was curious, you're, I'm sure, aware of the work that was done with the iPsych Broad Autism Group and the Psychiatric Genomics Consortium for Autism, where they looked at polygenic risk scores, but also looked at the burden of de novo damaging variants and the intersection of those two. And schizophrenia, they looked at schizophrenia as a comorbidity to autism to that point, but we know that they travel together because of developmental brain disorder. The question I had, and it was striking when you looked at the distribution because you had that tail, is how do we account for the fact that some of those individuals, if not perhaps the majority of those individuals that are out on the tail, actually have an unrecognized single variant over and above their polygenic risk score, a copy number variant or some other sort of a single variant, which we're not going to get out of genotype data, although we could get, if you had SNP data, you could get a copy number variant, presumably. And when we think about risk scores writ large, particularly when we see a fairly sizable burden of monogenic disease for traits like schizophrenia and autism and that, how do we account for that as we begin to apply polygenic risk scores?
I don't know if I've remembered all that, but with psychiatric disorders, the evidence for genetic architecture is that the sequencing studies of the same size have found variants for autism and very, very few for schizophrenia. And so there's a tendency, the paradigm model is that if you've got a de novo mutation of large effect, then you're having something that presents early in childhood, whereas the adult onset disorders are showing to be polygenic. And so I usually show a picture for this. The adult onset disorders are mostly polygenic. And of course there could be, as you say, a copy number variant, but those are very rare.
Unfortunately, I have data that you don't have that suggests they're way more common than we're thinking. Perhaps as much as 1% of well-characterized copy number variants are actually present in a population-based sequencing study. So that was unfair. But I think the point remains, because even looking at familial, like the Simon's collection, things like that, looking at, while it was primarily for autism, looking at schizophrenia, there's actually, I think, a higher burden. Perhaps not as high as autism, but still a reasonably high burden. And even if you're talking about, 3 to 5% of individuals within a population having a strong monogenic, I can't believe that it doesn't somehow confound how we interpret polygenic risk scores.
Well, I have seen some studies where essentially the evidence is, I think, that they're additive. So if you have something monogenic, then you need a lower burden of polygenic risk to cross a threshold. Yeah.
Thanks. I have two related points. One on sort of general discussion of clinical utility, and the other on specific statistical measure. We brushed the issue of the AUC under the carpet earlier this morning, and I'm going to bring it back out for just a second. But on the issue of general clinical utility, I think this goes to the sort of the threefold risk as being sort of the magic threshold. That we can identify this many people who are above that risk. I think those are nice calculations for sort of proof of principle, but now as we're thinking how this goes forward, we have to think very hard about the specific clinical context we'll be implementing these things. So I think in the risk stratification world, so if we're going to flag people as high risk, what's the intervention, what are the risks that come with that, what are the potential benefits, like those, calibrating that, to misuse the terminology a little bit, is the hard part, and will be very disease and question specific and context specific as well. So I think there's a lot of papers that are coming out that are sort of showing in principle we could do this, but the actual practice of, does it make sense or not, is the hard thing to sort out. And that sort of segs into the AUC. It's worth keeping in mind that the AUC is a measure that's taking the average sensitivity and specificity over the whole population into account. And 99% of the population may not be the people we're interested in. We really are interested in the people who are at the high-risk tail. So there's both a potential for overestimating the importance of a marker, but probably underestimating the importance, because it's really hard to change that AUC once you get above a certain threshold. And yet, the information that the additional marker is giving you will be very important for a small fraction of people out in the tail.
I have a question maybe for anyone in the group. I didn't really hear it addressed. How do you make the decision? Some of you were talking about PRSs that had over hundreds of thousands of variants in them, and some of them as few as 3. So how do you weigh whether it's good to have a PRS that's based on tons of SNPs versus just a few? Or does it not matter? As long as it validates, you don't care?
It really depends on the underlying architecture of the disease, right? If there's a highly polymorphic disease, in order to be predictive, you will need many, many variants, because they all have very small effects. So I think but if you have a disease that it's not as polygenic, then a few high-risk variants might be very predictive. So I think it really depends on the underlying genetic architecture.
I agree with that. It also depends on the sample size of the discovery sample. It's about being able to separate out those true positives from those false positives. And whereas with genome-wide association studies, we have that strict threshold where we only take those that are genome-wide significant because of the actions that we're going to do as we take those variants forward. With prediction, it doesn't matter that we take through some variants which aren't associated. It's about the proof is in the pudding. What is the prediction? And so, and the answer will be different for different diseases because of the architecture.
Is it a statistically arrived at number?
Well, there's different methodologies, and so there's some, and it does depend on the architecture. So it's definitely underpinned by hardcore statistical methods. But
sounds like Peter's got something to add.
Well, I think you're right. On some level, it's the proof of the pudding is in the eating, right? So if you will see whether you're doing better with 1 million markers or 100. So I agree with everything that Naomi and Monty just said. The one thing I might add is the sort of practicality and logistics of it. So if for whatever reason it's easier for a clinic to implement a polygenic risk score with 300 SNPs as opposed to 6 million, I'm not saying that's going to be true now or in the near future, but if there's not that much of a difference between the super high-powered, the whole damn genome versus these 300 variants, but you can do the 300 variants, then there might be a reason to lean towards that.
Steve?
I mean, I think this gets at a really interesting question about the balance between what do we want to do versus what do we understand. And when we understand PRSs or rare variants, there is a sensitivity and specificity to each of those. We know, I mean, a very elegant presentation earlier, 80-year-old person walking around with a CF mutation. These things are out there. And I know in Geisinger, I mean, in other databases, you see higher fractions of these highly penetrant mutations than what we have seen in other surveys, and we would expect in disease incidence of those sizes. So it really does raise this very important question of our bias in what we're trying to establish. Are we trying to push disease? There's a healthful side, there's a preventive side that we've barely talked about in terms of where, and it's a really hard thing to study, the genetics of not getting breast cancer. I don't know how to define that, but that's really important for the 88% of women who aren't going to get breast cancer. We know that 12% over the course of the next, in their lifespan. So, you have to look at the, sort of the severity of the disease. You look at Alzheimer's and take that to the extreme of Huntington's. Do you want to know that information at age 60? I was sitting here, I'm 63, saying, no, I don't want to do that math test. I don't want to have that PRS. I have a family history. I don't want to know that at this point. So context really does matter. I mean, I think the numbers are critical, but one of the hard things is where we're going to put this information in the context of using it. When do we assess it, and when do we act on it, and when do we not act on it? And I think there's some good examples where we should act, but there are plenty of them where we don't know enough, and it may be more dangerous to say things than to not say things. And that's a difficult thing as a physician, to be in that position.
So I guess my question is for the statistical geneticists in the room, if they could address the issue of additivity and multiplicativity. So I mean, if you're taking a score based on the log odds ratios and making a sum over that, then you're talking about multiplicative effects, I guess, on the raw scale. And what are the expectations of that for the deviation and the extremes of the distribution? So why is it, for example, in Alzheimer's, you were getting that top decile with a massive increase and nothing else? What are the laws that we can use to predict how much of an extension you expect under additivity and multiplicative effects?
So it's additive on the underlying scale, and as you say, multiplicative on the disease scale, and those shapes of those graphs are exactly what you expect. You know, I can derive those just from the variance explained by the predictor. And
But
yeah.
Is there any possibility of actually precisely comparing those slopes at the ends and getting information on non-additive effects? I mean, they are what you expect in terms of their S-shaped, but is there more information in there?
Well, I don't know, other people might want to say, but I mean, from my perspective, it seems that when effect sizes are so small, that difference between additive and multiplicative, you can't differentiate. So with polygenicity, it's additive.
Carol, next.
Yes, I was actually also, in terms of the number of SNPs, and I don't know if this is the right way to think about it or not, but one of the things I think about as you get to the larger numbers, relying on imputation rate, and then you also start thinking about how well is what I find in one population going to transfer to another group or another area. And so I guess, like, is that extra precision that you're getting from going to the larger numbers sending you more specifically into whatever population you're studying or whatever population you're sort of imputing into, or is that not a fair way to think of it? That's the question.
Yeah. I think that's a good question. Anybody want to address it?
It looks like Peter's going to answer it, but I can.
Oh, well,
Let's let one of our...
Yeah, I would say two things. One is that in terms of the question about...
Use the microphone, please.
In terms of the question about about is more variance better, I think we've had a number of good points, which really you just got to check and see. And even in our work and a number of other groups have looked at it, and even in the external samples where they may have used different variants and different imputation reference panels, in the work I'm familiar with, the larger scores do capture a greater proportion of a variance and have a greater effect size. So at least the empiric evidence suggests that it is useful to go to that expanded number. And then the other question that came up was about the whether or not there was a nonlinear relationship. And so at least in our work, we have not found real evidence for that. I think so in some of those curves, like in the paper that's in this pamphlet, it looks like there's an S shape when folks are plotted on the x-axis is the percentile and the y-axis is just the prevalence of disease. To me, that was like the most intuitive way to present the data. But you can actually make that curve a straight line. One, by making the y-axis the logit function, so log odds of p over 1 minus p, I guess, and then the x-axis not percentile bins, but actually just the raw score. And so I think somebody else mentioned that really, if you have just one or two parameters about the score, variance explained or odds ratio per standard deviation, you can actually derive the expectations. And in our cases, they've actually fit quite well with those expectations. And so we're working now on different modeling to say, if you just know that one number, to what extent can you use various, for example, threshold liability models to figure out how someone might cross over in a health transition from healthy healthy to disease. But it's a long way of saying we haven't seen a lot of evidence for non-additive effects.
Other comments or?
Can I just ask a question? I'm not a geneticist. I'm just wondering whether one could kind of increase the kind of predictability of polygenic risk scores by using intermediate phenotypes. So for instance, we talked about tapping the electronic health records, and we know that clinical risk has so much environmental exposures, but we know that also, for instance, total white cell count has been associated with high mortality, and different intermediates that can be measured in the electronic health records. And each of these is a complex trait. So if you can just take the kind of risk factors associated with these and build them all into a kind of more definable polygenic risk score. And you're probably very naive. I'm not a geneticist. I'm just asking what I thought, something got obvious to me anyway.
Yeah, so this actually reflects back to a question this morning that when we ran out of discussion time, I can't remember who exactly put it on the table, but it was about the idea of outcomes. And when we were talking initially about outcomes, a lot of the outcomes that we capture are the process outcomes, which is did somebody do something or did they not do it? But you're correct that there are intermediate outcomes that we use in clinical care, like the LDL level in an individual with a lipid disorder. We know that if we can get them to a target, there's strong evidence to suggest that we are going to have a health impact, even though the health impact may not show up for 20, 30 years. I mean, if you're doing FH in a kid and you're starting statins at age 8, you're going to need 50, 60 years to know whether or not you had a true impact on the health outcomes. So you have to have those intermediate outcomes. So we've actually defined outcomes related to many of these conditions that are related to monogenic disease, so whether it's familial hypercholesterolemia or hereditary breast and ovarian cancer. And so I think one of the interesting questions with polygenic risk score are the outcomes that we've defined for monogenic disease the right ones to measure for polygenic risk scores? And if they are, great, but if they're not, then what do we need to fill in the gaps? Because you're absolutely correct. If we want to really learn whether we're having impact and measure clinical utility, we have to have outcomes with evidence to know that what we're measuring actually has the ability to predict the health status down in the future.
I could maybe talk to that one as well. So, I mean, I think you're right. We can take genetically correlated traits and make a predictor which use the information from correlated traits to make an improved predictor. But in the case of LDL, you might do that, make that predictor early on in life, but as someone progresses through life, then you take that actual LDL. So you'd make the predictors changing over time. The other point I can address is about increasing sample size. So we often talk about when do we need to stop increasing sample size for discovery in genome-wide association studies, when will we stop discovering variants? But when we're talking about prediction, we need to keep increasing the sample sizes, because what we're now interested in is the accurate estimation of the effect size of each SNP. And also, as you say, as you increase sample size, what we want is more phenotypic information, and that will then also help break down heterogeneity, because one of the slides I showed was how psychiatric disorders are shown to be very polygenic, many more small effects compared to other disorders. And one way to interpret that is maybe that's a feature of the disease, or maybe it's the fact that we've compacted together different diseases because we haven't been able to separate them yet because our diagnoses aren't that good. So large sample sizes will do many things.
Steve, and then.
Yeah, I mean, going back, relating the last 2 points, I think, there is this whole world of Mendelian randomization where you have the model of a disease and an intermediate risk factor, and the question is the genetics of the contribution to those risk factors that some, like Richard Holston, has dubbed as finding what you already know, the degree of genetic prediction of obesity for going on to type 2 diabetes or colon cancer. But I think there are some very interesting things starting to emerge where you break them apart and see what's in, for instance, a BMI or obesity as separate from colon cancer, and asking the question, can you in some way begin to work those together? Breast cancer, we know BMI is a small but important risk factor as well for postmenopausal, not so much for premenopausal, so you have to start to look at the subtypes. But I think there is light at the end of the tunnel for starting to look at some of these intermediate outcomes in more creative ways that would combine some of these kinds of observations. Not that you will change the AUC or the R squared that much, but it's nonetheless, I think, a very important aspect, and one that may have some public health implications if you indeed see that there's a part of the population that's at particularly high risk because of obesity for a particular disease. But it's going to take very large sample sizes to work those things out, and that's why, I mean, I agree with Naomi. You stop scanning when you no longer find variants that are associated with that disease, and as we have more subtyping and more of these refined features, we will find that it's more and more challenging to get those numbers, but we have to get them.
I've got George, and then Dan, and then Nancy.
Thank you. This is a question for Patrick on the atrial fibrillation. If I recall correctly, one of the things we learned from the early Framingham data was that in young adults who didn't have hypertension or diabetes or structural heart disease, AFib didn't carry as bad a prognosis as those who had structural disease. Are your data suggesting, or are you finding that there's a subset in the lone atrial fibrillation in whom PRS might suggest a higher risk and therefore be managed aggressively? And also, based on the discussion this morning, do you find in that group a truly low-risk population of young AFibers who we should leave alone?
Great question. We don't know yet is the simple version, mainly because of the, I think the initial risks, if you have isolated AFib, the question is, do you have a low risk for, and you're young, do you have a low risk for stroke? We don't have great data to resolve that. I mean, the CHADS, VASC 0 and 1, and that's wearing more of my clinical hat than someone doing genetics. That low risk population, the CHADS, VASC score is not based on huge amounts of data for that ascertainment of risk. And if you look at the ST statistic or something for CHADS, VASC, it's pretty lousy. It's 0.68 to 0.7. So it's not very good for trying to do that. And we know that there are still events in that population. So unfortunately, the challenge is that the number of patients with that isolated AFib at a young age are small, the event rate's low, and therefore the number of people that you would need to enroll in a study is huge. And so because of that, I don't think we're going to have any resolution on this answer anytime soon. Does that make sense, sir? Yeah, thanks.
Dan?
Hesitant to ask this question, but because I'm not sure when the right time is, but so I'm going to wear my clinician hat for a sec. I have somebody. Stop it! I have somebody in my office. Yeah. Okay. I'm going to sit on my hand. I have somebody in my office who brings me their Amit Kara genetic risk score for atrial fibrillation, and they have a risk of 10. They're in the top 0.01 percentile, and there's a whole bunch of things I can do for them or to them or whatever. But the question is, then they asked me the question: should my brother see you as well? So what is the family relationship of a high genetic risk score? If I have a high genetic risk score, does that mean my first degree relative also has to have a high? I mean, this is like a Mendelian question. If they come in with a BRCA1 variant, I know what to do. But the genetic risk score, and I've thought a lot about how do you get to a high genetic risk score? Your parents both have to have a high burden of homozygosity at least some of those sites, so you're likely to have a higher than average risk score. But is your sibling going to regress to the mean? Or so what do I tell how do I do family counseling to people who have high genetic risk scores? As high as you want to say as monogenic disease. And again, I don't want to get into that debate, but I disagree with that statement. But I want to know about what do you do about counseling in 2030?
Yeah, I would say the
Microphone.
I just to distill that question a little bit more, which is basically what is the distribution of polygenic scores among offspring, let's say of a set of parents, and if
You'd actually have to have data. I'm working in a data-free zone. I'm doing simulations.
well, I guess it's a call for data, which is if my score is 0 and my wife's score is 1, on average my daughter's score is going to be 0.5. But I think there's a lot of trio data available, and available. Just say what is the spread of that distribution? Like, is it everyone's between 0.49 and 0.51, or is it a spread of distribution between 0.3 and 0.7, for example? And so my guess is actually a pretty narrow. It's a pretty narrow distribution, but it's not something I've seen. I personally have seen direct data.
Well, if you have 6 million SNPs, I guess it would have to be narrow.
Doesn't that depend how many SNPs?
And if you have 150 SNPs, it might not be so narrow. And the parent-child, this is like an idiotic question. Do parent-child trios actually help, or do you need parent-child sib-sib, I think, or the more the merrier, right? I mean,
So under a polygenic model, half of the genetic variation in the population is actually within a family. So a pair of parents, the variation that they generate is half the genetic variance in a population. So there is a lot of, relatively a lot of variance about the mean of the parents. And that's why polygenic risk prediction is good, because within people with a family history, it can also help to differentiate between people.
I think you just said more elegantly what I was trying to say, and that is sort of that in order for me to be in the tail end, I have to have, my parents have to be homozygous at some of those variants.
No, under polygenicity,
Or am I
it's
wrong?
really about a count.
Right.
It's not thinking about homozygosity at loci. It's about the cumulative count. So
Just
Okay.
one copy's enough, right? If you're lucky.
Right,
it's
no,
the
no,
count across many loci.
But across many loci, if you have only one copy, your genetic risk score will go down then, compared to if you have 2 copies at a particular. Am I getting this right?
But being a homozygote at many loci is like having, throwing the coin as, heads all along the way. But it's just saying a high polygenic risk score is saying you've just, across all those throws, you've got a higher number of heads.
Nancy, do you still have a question?
It was back to a comment that Naomi made. I think it is really important to recognize that we will learn more and more about shared genetic architecture and pleiotropy, and that should improve the ability to do prediction in a number of ways. So if you think of something like Alzheimer's, as we understand more about the phenotypic signature left behind in the electronic health record by genes as they fail on their way to Alzheimer's, that phenome can be collected. You will recognize it as having shared genetic architecture, and you can build larger targets then for the predictive models. And as we go forward, really prediction should get better and better, because we'll understand more aspects of the total architecture and use it. That's the key, is to use it when we find it.
Yeah, I was actually thinking about the same question that Dan just had. It's a great question, and because it has implications for cascade testing. So we know what to do with the monogenic conditions because you have a 50% transmission rate, but I think it would be interesting to do some simulations and see what kind of effect it might have on detecting other relatives in the family with high scores, and what might be the distribution of those scores based on high polygenic score in one member of the family.
So as Naomi pointed out, this is Fisher 1918. Like we know this. If you know the variance of the polygenic risk score in the population, you know what the distribution is in your relatives, conditional on your score.
And genetic counselors have had this on their exams since the exams began. This is not, so it may be that physicians have internalized some of the Mendelian genetics and are comfortable with those, but this has been used in genetic counseling situations since exams began. So yes.
Dan, I would stop with the first part of your question, which is, say you have someone at that 1% risk, what are you doing for them? Right, I mean, before you even get to the screening of the family or anything else, what about just that individual in front of you?
Well, for atrial fibrillation, nothing, but maybe for BRCA, maybe for breast cancer, you might institute earlier screening. I mean, that's.
But even for AF, I mean, what, doesn't it depend a lot on the substrate? I mean, if you're 65 and you're at the top 1%, would you wear a watch?
Yeah, this comes back to our discussion over at the bar last night. But I mean, so we'll pass on that. But I mean, yeah. I mean, I think that at some point, so you're talking about increased surveillance.
Sure. I mean, it depends on the extent of the. I mean, I'm not suggesting injecting a monitor into everyone at the top 1%, but if you have a surveillance technique that's straightforward and safe, and it's actionable when you get a result. Okay. You know, in the over 65 range, if you detect AFib, you're going to be anticoagulated, almost all of the patients. So.
But I still don't know what to tell my brother.
I don't know what to tell your brother either, but he may not have a very high risk.
We haven't talked much about the non-European risk groups, and so almost everything that we heard was from European ancestry populations, understandably why, because that's where the data are. But as we as an institute try to move forward, we don't want to just move forward in European ancestry populations. And so recognizing that these don't predict as well in non-European, do they predict well enough that they can be used? Are there obviously caveats, but also, you know, ways that we can adapt or modify or whatever to account for the fact that allele frequencies are different and when, you know, prevalences and risks are different.
Just add on to that. So don't you do better with a PRS that's based on hundreds of thousands of SNPs predicting across populations than one that's based on 20? Or no?
Yeah, so starting with your question, like what can we do? Right now when we're looking at our vast Eurocentric study biases and trying to see how well our scores work across different populations, we're doing a pretty abysmal job, especially in African descent populations. The more genetically diverged you get, the worse you do. And so I think in terms of thinking about how we can have a polygenic score that actually benefits more than just the white subset of the population that's already on the like positive end of health disparities, we like vastly need to shift our study priorities, I think, if we want to have any hope of benefiting all in sort of an equitable sense. So that's both an ethical and a scientific issue, of course. And then to your question about the like number of SNPs, this also kind of gets back to the questions of portability across populations. As you start adding in more and more SNPs, we see a little lower and lower generalizability, and that becomes, yeah, so as you start to dip into those, you're making this trade-off basically between signal and noise. So at the top end of your like GWAS studies, you have mostly signal. As you start dipping lower and lower into your GWAS summary statistics, you're getting a little bit more noise, and that noise is tracking with your population a little bit better. And this becomes problematic even within the populations that we already have very well studied. So for example, we have a study in Finland. Finland is a really useful population to study for all sorts of translational, like this might be like the leading edge of where we might think about polygenic risk scores translating. But like already within Finland, there's massive amounts of stratification and there's over-predicted differences geographically. So like we're kind of at the starting edge, I think, of like understanding how that the number of SNPs is going to impact geographical differences across populations and what this means for translation across different populations.
Mark?
I was just thinking that, this would be a really, given that we have a number of polygenic risk scores that have been validated, and given that we have some really nice phenotyping algorithms that have been validated against longitudinal healthcare data, I could see a relatively easy synthesis for the large number of groups that have both the genomic data and the electronic health record data to basically just throw them out there and say, what's the performance characteristics based on what we know. Now, again, the extant ones are still relatively underrepresented for a lot of the race and ethnicities, but I think you might be able to, get to a relatively small group probably of ones that actually perform reasonably well across a wide range of populations where you find others that are quite disparate, and that might be important in terms of thinking about how do we prioritize implementation.
So it seems to me that there's an implicit assumption that once you have a causal variant, it has the same effect in all groups. At least that's what I've been hearing today, and there's certainly been papers saying that. So I think it partly, our response to your question, Terry, partly depends on what we think about that assumption. Do we think it's correct or not? If it's not correct, clearly we need to go to different ethnicities and get their own polygenic risk scores, which of course is an expensive gathering, but necessary. If we think that they are the same across populations, that means we need to get to the causal variants, which may also be a very expensive project. But that's what it seems to me.
And if I could just say, I was sort of asking the question, what do we do now? So granted, those are the two things we'd really like to do in the future, and hopefully we will. But what do we do now?
But I think we already know from genetics and model organisms that, in fact, a causative variant in one individual is not necessarily causative in somebody that's in a different background. So I think it's really important to, I've heard several times people talking about causative variant being a causative variant, and I think we need to think about the impact of other variants that are in a genome that might be different between different individuals. But the model organism world is very clear that just because you have a causative variant in one background, it will not be causative in another.
Yeah, but there's a lot of examples to the contrary. In pharmacogenomics, for example, every actionable variant is actionable no matter what race group it occurs in, essentially. So
right. It's all about what the other genes that are intersecting with the quote unquote causative variant, and so we have no clue about 99% of them. Yes, in pharmacogenomics we've looked at 50 or less.
So I'm sorry, but we're going to have to hold this off. So I'm sorry to have raised that question in the last 5 minutes of the discussion. But please hold your thoughts. We do want to get to the debate. One of our debaters has a White House function to go to, and so I know, so we can't really. So could I? It's only the White House, really. So could we come back at 4:35? So that's 14 minutes for a break. But please be back 4:35, and we'll get started.